This is Chapter 4, Video 2, relating to Section 4.2 in your textbook. It's about genes and how they work in the body. We're going to review the definition of a gene and genome and talk about why the genome is important in the health science fields. We'll talk about the genetic code and how DNA codes for proteins. We'll talk about how amino acids are formed into proteins. We'll talk about what happens to a protein after we have the basic sequence of amino acids established, and we'll end by talking about how genes can be regulated or turned on and off. But first, this is a very common problem that you're pretty likely to see on an exam, so I wanted to give a quick example of how to think your way through it. Here's how a question might read. A portion of DNA is composed of 35% thymine. What percentage of that portion of DNA would be made of guanine? It's really interesting that if we know the percentage of any one of the 4 nitrogenous bases, we can figure out the percentage of any of the other 3. So let's look at how this would be done. We know that A and T are always paired together, so we're going to have equal amounts of them. If I have 35% T, as we do in this problem, we'll also have 35% A. And that means 70% of our DNA would be made up of A and T. If 70% of our DNA is A and T, that means 30% must be C and G. We know that C and G always pair together, so this 30% must be evenly split between C and G. So if we do 30 divided by 2, we'll find that 15% of the DNA is G, which answers our question, and the other 15% will be C. To review, a gene is a segment of DNA that gives directions for making one particular protein. Humans have about 20,000 genes. The nucleotide sequence that a DNA molecule has gives us the amino acid sequence that the protein will have. If we consider the entire set of DNA that a given person has, that is that person's genome. And genomics is the study of the genome and how the genes and the non-coding DNA, which remember makes up a very large percent of the DNA, this non-coding portion, how they work together to impact the structure and the function of the whole organism. A single gene can actually give us several different proteins, and we'll talk in a minute about how that's possible. The average gene is about 3,000 base pairs long, though they can be much shorter or much longer. Humans share 99.99% of genetic material with each other. So all humans are extremely similar in their genetic makeup. However, 2 individuals can have up to 3 million base pairs different and still fall within this 99.99% similarity. That's due to the incredible length of the DNA and the huge number of base pairs that make up our DNA altogether. There are some disorders that are caused by one nucleotide being wrong in a person. We have 23 pairs of chromosomes. So of those 23 chromosomes, some of them carry a lot of genes and some of them don't carry all that many genes. So in reality, some chromosomes are more important than other chromosomes. We know the exact location of 1,400 different mutations that can cause diseases. And this allows us to some personalized medicine options where we target specific places on the genome and look to fix mutations that have happened at specific places. If we know that a person has specific mutations, we can do a better job at predicting their disease outcomes. We can diagnose diseases sooner, even sometimes before symptoms are severe, and we can also treat diseases more specifically. The whole library of proteins that a human makes is called the proteome. So all of the proteins that a person makes all together, the proteome, are all made of just 20 different amino acids in various sequences, and then those sequences take on various specific shapes. Those 20 amino acids are coded for by just 4 nucleotides, and this is called the genetic code. This is the system where we have a very simple DNA code with 4 variations that gives us a slightly more complex amino acid code with 20 that can work together to make millions of different proteins. So this happens by a process where we look at base pair triplets. Okay, so a base pair triplet is a sequence of 3 DNA nucleotides, the A's, the C's, the T's, and the G's. And a set of 3 of those nucleotides, using the code that we see here, gets translated into 1 amino acid that gets added to a growing polypeptide. Okay, so when we look at the first position, okay, so our triplet, for instance, let's look at here. This triplet, AUG. So my first nucleotide is A. So in my first position, I have an A. Second position is U. Okay, so U is going to be here. So that means first A, second U. I'm looking in this box here. And then third is G. Okay, so I'm going to look at third position here, G, and I slide over. And that means that depending on if it's the first thing in the sequence or if it's in the middle of the sequence, this is either my start codon or a methionine that's going to get added as an amino acid. So the things here in the middle of the chart are the names of the different 20 amino acids, as well as our 3 codons that mean stop. Okay, so remember that the amino acids, there are 20 of them, and we know the identity of the amino acid by what R group is attached to the central carbon of the amino acid. Okay, so your body knows which amino acid to add based on the DNA and then mRNA sequence that it encounters. And we're going to talk about how this works in just a moment. All our cells have the complete DNA molecule. So that means they all have identical genes, but different genes are turned on or activated in different cells. The rest of the genes aren't used. They may be dormant in those cells if they aren't being used. So let's look at how this happens. Guys, the stuff that's in this blue box here is the absolute most important thing out of this whole chapter. If you truly understand what's in this blue box and how this happens, you will be 1/4 ready for this exam, because exam 1 is about 4 chapters. This blue box is absolutely critical information. You need to understand the processes that are being talked about here. Okay, so here's the process of protein synthesis. We start with the DNA molecule. From the DNA molecule, we make an mRNA template, and that mRNA template is translated into a protein. So this first step, DNA to mRNA, this is transcription. Transcription, where DNA makes an mRNA, happens in the nucleus. Translation, where mRNA codes for a protein, this happens in the cytoplasm. Okay, so we have transcription followed by translation. Transcription happens in the nucleus. Translation happens in the cytoplasm. So transcription is how DNA is made into an mRNA code. There's an enzyme involved here. Remember, our enzymes end in ASE most of the time. So RNA polymerase is our enzyme that makes mRNA based on the DNA. When we're making mRNA, every time that the RNA polymerase finds a C, a G gets added. C and G go together. Every time the RNA polymerase finds a G, a C gets added to the mRNA. Whenever we find a T, the RNA polymerase adds an A, and then this is the one that's a little bit different. We don't have T's in mRNA, so when we find an A, we add a U or a uracil instead. So our mRNA is made up of C's and G's, A's and U's. So our DNA, the particular section that we're interested in, gets made into an mRNA. And our mRNA has some sections called exons. They're here in the light pink, A, B, C, D, E, F. And also introns, which are the dark pink. And the introns can be cut out. And then the exons can be arranged in different orders to make different proteins. So if we were to put A, C, and D together, so this section, and then we're going to cut out all of this. We're going to take the C, we'll cut out this intron, and then take the D. That's going to make one protein. Over here, if we are concerned about A, E, and F to make a different protein, we would take the A, and then we would cut out all of this section here in the middle, and then we'd take the exon for E, leave out this intron, and take this exon F. And this would give us a totally different protein than our protein that we have over here. All right, so now we're getting into translation. This is where mRNA gets made into the amino acid chain, which is the beginning of the protein. Okay, so translation has three things necessary. The first one is that mRNA that's carrying the DNA's code. Then we need transfer RNA, and we've talked about this in early videos. Transfer RNA is a very small molecule that's carrying one single amino acid. It's like a little wheelbarrow that's carrying one amino acid, and it's going to make sure that the right amino acid gets added to the growing chain. And then the last thing that we need is the ribosome. And you guys are going to remember from chapter 3 that ribosomes are the organelles that build the proteins. So they're the organelles that are going to read the mDNA or the mRNA and are going to make the protein. So mRNA, we talked about this. mRNA is the code. It starts with an AUG, and then each three letters, each three base pairs are going to add a new amino acid. tRNA, here it is. It has a place where the amino acid gets tagged on, and then it has a region called the anticodon, and that's going to match up on the ribosome to make sure that we're adding the right amino acid at the right time. Translation, remember, this is the process of turning mRNA into an amino acid, which will then be turned into a protein, happens in 3 steps. Okay, the first of those steps is called initiation, and initiation happens when mRNA attaches to the ribosome, and we have the first start codon matching up with methionine, which is carried by the tRNA. Then we have elongation, and in elongation, a whole series of tRNAs bring the next amino acid in the sequence to get added to the building protein. And finally, when we get to a stop codon, we're going to have termination, and this is where the protein is done being made. No more tRNAs come in carrying amino acids, and the amino acid chain is complete. This translation, using the mRNA code to make the protein, can happen very, very fast. Sometimes we can have over 100,000 proteins being made per second. Okay, so this can be a very, very fast process. After translation is done, after that amino acid sequence is finalized, there are still some steps that have to take place. Okay, remember that the amino acid sequence is just the primary structure of the protein. We have to fold it with the alpha helixes and beta pleated sheets, and then into its three-dimensional structure. And those happen starting in the rough ER. So the newly made protein, that sequence of amino acids, gets sent to the rough ER where it's modified and packaged. It's put into a transport vesicle, kind of like a transport bubble. And we can see just a quick review. So we're starting with the DNA. And then we have the mRNA that's made from the DNA. And then we have the tRNA that are carrying our amino acids. And these amino acids get put together in the right order. And that's how we get the polypeptide. So once the protein is assembled, then we have to make sure it gets its three-dimensional shape. So this is done by the endoplasmic reticulum, the ER. And this happens in the rough ER once that protein is in the right sequence. Okay, so it gets folded, and then the different bonding that takes place on the inside happens, and some carbohydrate components may also be added. And then we have a complete protein. That complete protein goes into a vesicle, which is like a bubble, and that's going to transport it. And it's going to get transported to the Golgi, which is another organelle that we've talked about previously. Okay, so the Golgi is going to help that protein mature a bit more. Some different carbohydrates can be added to the surface. And now, with a completely finished protein, the Golgi is going to either store it or ship it to the location in the cell or in the body where it's going to get used. The last thing that we're going to talk about here is gene regulation. Gene regulation is about how genes can be turned on or off as they're being needed or not needed. So, some proteins are only needed in certain places in the body. Okay, so hemoglobin, for instance, a big component of your red blood cells, your liver doesn't need that. So, it can be turned off entirely. Another one that is important, some of the proteins in breast milk, for instance. Casein is one of those proteins. Men don't ever need that, and women only need that when they're lactating. Okay, so we can turn off that gene. There's no need to make it unless it's needed by the body.